I need to provide a comment before we start. This is, for those of you who don't have a background in neuroscience, there's going to be some challenging material, which means that anyone who took my picture may want to erase my picture from their phone, because after this talk, you may never want to see me again because of some of the material. So here's what I'm going to cover in my presentation today. How does early, profound psychosocial deprivation impact brain development and biological development? Can these effects be reversed? And if so, is the degree of recovery influenced by how old the child was when they were removed from the institution and put into foster care? So I'll start by talking about the effects of profound neglect on brain development. Then I'll talk about how psychosocial deprivation impacts brain development, focusing in particular on EEG and on MRI. I'll talk about how psychosocial deprivation impacts this part of the stress response system. You heard already some of this from Professor Fox. I'll talk about a few additional findings. And then how psychosocial deprivation impacts biological development. And specifically, I'm going to focus on something called telomere erosion. And don't worry, I'll define telomeres in a little bit. So we went through a little bit of this yesterday. The question is, why is neglect bad for the brain? So the brain expects input it doesn't receive, so its wiring is altered. So the way I want to illustrate this is that when we talked yesterday, we discussed how the brain, much of the brain after birth depends on experience. So the way to think of this is that the brain is sitting there, genes have made some brain circuits and made brain cells, but it's waiting for a set of instructions to tell it how to develop. And those instructions are carried by the environment. So after birth, the brain is sitting around saying, okay, this aspect of experience, this experience will all tell my brain how to assemble itself together. The worry we have is that if the brain is neglected, it's not getting those instructions. So it would be as though you're sitting here waiting for me to talk, but I don't say anything, and therefore you don't learn anything. So brain wiring is based on experience, and the concern is what happens when there are no experiences because the brain is deprived. This is particularly egregious, as we also discussed yesterday, if the experiences that are supposed to occur during a critical period don't occur, as you heard in the previous talk. So the question is, what happens to the brain when it's deprived of most expectable experiences like caregiving, high-quality caregiving, and patterned light and language input and things like that during a critical period? So what I'm going to do today is draw from our work on EEG, magnetic resonance imaging, a little bit about the autonomic nervous system and stress, you heard some of that already, and then biological development, telomere erosion in particular. So this slide here illustrates a number of different ways we can record brain activity from young babies. Starting from the top, we have EEG. In the middle, we have what's called magnetic encephalography. Don't worry, I won't be talking about this. We have over here functional MRI, which we will talk a little bit about. And then down here we have something called near-infrared spectroscopy, another tool that is being used in Porto Alegre and also in Salvador that's focused on the hemodynamic response. So let's start with, what is the EEG? So the EEG reflects the synchronous activity of millions of neurons distributed in different parts of the brain, and these neurons work as a unit all together, and that activity propagates to the top of the head, to the scalp surface where we can record those that activity. So a metaphor here might be in an earthquake. So far, far away, there's geological activity that gradually rises to the surface of the earth and splits the earth open, what we call an earthquake. But the origin of that earthquake, when the earth opens up, actually could have occurred hundreds or thousands of miles away. So our challenge is much like what a geologist does. We place sensors on the head, and we're listening to the original source of of those millions of neurons that are working together. And we don't need to know this too much, but there's a particular type of neuron called a pyramidal neuron that really is what generates these brain currents. The next thing to know is that the EEG is a multidimensional signal. So we see changes in voltage over time. If you look over here, you can see these squiggly lines, all right? That's the EEG. So if I put electrodes on your head right now, I would be recording your EEG from throughout your brain, and it is manifested in squiggly lines. It's also represented in space. So what you're looking at in these little circles is where we place each one of our sensors. So imagine looking from the top down, and we have 128 of these sensors distributed across the scalp. So we have space, we have frequency, and so the way to think of this is that some brain activity is very, very fast, some is very, very slow. So right now, for those of you who are sleeping through my talk, I would see this. For those of you who are not alive, it would be down here. It would just be a flat line. I'm hoping that you're showing activity a little bit like this, slightly faster activity. We can look at the power. Think of this as using a metaphor for a light bulb. If you turn the brightness up or down, EEG power reflects more brain activity or less brain activity. And here's another way to view this. So this is the frequency domain. The very top would be excited or relaxed versus drowsy versus asleep, deep sleep, and then finally all the way down to if you were in a coma. Now, the next slide will be a little bit alarming, but here's what I'm trying to explain here. This, we can work out at a very, very molecular level what's going on as different types of cells communicate with one another. But even better is that we can connect this so that what we can do is go from the deeply molecular level and then on the other end actually look at how healthy a child's development has been based on the different types of brain activity I've just talked about. So how do we record brain activity? Well, here are some pictures. You can have up here is a system we used many years ago when we were working in the neonatal intensive care unit. This is an old system where we can put on a small number of electrodes. But what you can see down here is the more modern way of doing this, where we can put on 128 sensors inside of 2 or 3 minutes. So it goes very, very fast. And if this is a movie, and I'm wondering if you can start the movie. This will actually show you what it looks like. She's sneaking up on the child. An undergraduate who's paying a lot of money to go to Harvard is sitting on this side, keeping the baby busy so she's not aware of what's going on behind her. And that's it. As you know, babies have very, very short-term memories. So the baby initially thought, oh, something's going on in my head. And then the baby said, oh, there's my foot. And is completely oblivious to this. So this goes on really quickly. So what we've done, we can record the EEG under what we call resting conditions, when nothing is going on at all. And in our case, when they were babies, I don't know if they have bingo in Brazil, but bingo is this game where you have a cage and there's little balls. Okay, so you know what this is. Well, it turns out babies like to watch those balls bounce in the cage. And you may as well, but I know babies do that. And it's something that entrains babies. Right now, I think in Professor Fox's lab and my lab, we might show attractive screensavers on a screen to keep a baby busy. Because as you know, they have very, very short attention spans, so it's only so long you can keep their attention. When the children are older, like 8 or 16 or 12, we can test them the way we would test you. We might say to you, keep your eyes open, keep your eyes closed, or put the lights in the room on, put the lights in the room off. And during that time, we can record the brain activity. So I'm going to start with our baseline data. This is before randomization. This is when all 136 infants are in the institution. On average, they're around 22 months of age. And what you're looking at here is at the top, you're looking at the institutional group, and at the bottom, you're looking at the never institutional group, right? So this is before randomization. You're looking at the top down, so right ear, bottom of the head, back of the head, left ear, all right? So you're looking at the top, and what we can do is look at the distribution of electrical activity across the surface and measure how much activity is there. And what's readily apparent is that as you move to the red spectrum, you're seeing much more brain activity. For the never institutional group, this is all sitting over the frontal lobe. Look how much more brain activity there is here versus in the institutional group. So it's as though you've turned the dimmer switch down on the light, and now you're producing less brain activity. Now, let's jump to the after randomization has occurred. This is 8 years later. The children are now 8. What you see in the upper left is the institutionalized group, the care as usual group. And in the upper right, you see children placed in foster care after 24 months of age. And those two are identical. In the lower left, you see the children placed in foster care before 24 months. And over here are the children who've never been in an institution. And they're identical. So what that's showing us is that placement before 2 leads to what looks like a normalization of brain activity, whereas placement after 2, those children look no different than the never institutionalized children. So let's jump to age 12 and plot it differently. But if you look at the area that I've highlighted, the red is foster care, the green is never institutionalized. And both of those groups are, first, they're identical. Second, they both show more brain activity than the institutionalized group. And as you heard, I think yesterday, by the time children were 12, probably half of them have already left the institution. This is based on intent to treat. So 12 years later, we're still seeing an intervention effect. And now if we go to age 16, we still see the same thing. This is foster care here, and here's never institutionalized, and look how much lower the brain activity is in the institutionalized group. So what that's telling us is that even at the age of 16, we're still seeing the effects of those early months or years, in some cases, of neglect. Then this is showing the effects of timing. What you'll notice in this, what's called a regression curve, is that on the y-axis, as you go from 1 to 6, that's the amount of brain activity. You'll notice, and on the x-axis is the age at placement into foster care. And you'll notice that the older the child was when they were placed, the greater the reduction in brain activity. And this is also at 16 years. So what that's telling us is that, as we saw earlier meaning 8 years, and then again at 12 years, and now at 16, children in foster care have more brain activity than children in the institutional group, and those placed in foster care before 2 have more brain activity than those placed after 2. And that's the critical period that we talked about. The last thing is I have to first introduce this concept, and Professor Zena will talk some more about this. One of the things that we've looked at is the number of placement disruptions. For those of you who work in foster care know that the more foster care homes a child goes to, the worse their developmental outcomes. Stability is really, really important. So we were able to look at our sample based on, in the foster care sample, Some of them, it turned out, were in very stable placements. After randomization, they went to one of our foster care homes and they stayed there forever. But there are other kids who bounced around. So for example, you go to our foster care, but then the authorities a year later decide to reunite you with your biological family. And then a year later, that family re-abandons you to an institution. And then you go to government foster care. And you know this pattern because it exists worldwide in other contexts. So we divided the sample into disrupted placements or stable placements. And what you can see here, focus particularly on the middle plot, the amount of brain activity declines as a function of placement disruptions. So we've already said that there's less brain activity with older placement. This is also showing that kids who have many placements show a reduction in brain activity. So to summarize, children who received care as usual, the institutional group, display deficits in brain activity compared to children randomized to foster care. Children who received the foster care intervention continued to show normative levels of brain activity all the way through age 16. In other words, there was no washout. The age of placement into foster care was associated with increased alpha power, earlier placement, more alpha power. And finally, more disruptions led to less brain activity. All right, so now, what about other measures of brain development? And now you need to brace yourself. Put your seatbelt on, because we're going to have to talk about physics for just a few minutes. So what we wanted to do was do magnetic resonance imaging on our sample. And here on the left is an MRI scanner, which will look familiar to some of you. And on the right is a view of the brain this way. So it's a sagittal view of the left hemisphere. And just to orient you, so here's the nose, here's the eye, here's the frontal lobe here. This is your cerebellum, here's your visual cortex, and all of this through here will be your spinal cord or your midbrain, things like that. The question is, how do you get those images? So a brief digression on MR physics. So magnetic resonance imaging uses magnetic fields to image biological tissue. Here's a 3 Tesla scanner, and this is the Earth's magnetic field. So Tesla was the physicist who, it's named after him. It's a measurement of how powerful the magnetic field is. You'll notice that the Earth's magnetic field is 0.00003 Tesla, whereas this MRI scanner is 3 Tesla. So it's, thousands of times more powerful. Typically, when we talk about MRI, we're referring to a signal that comes from hydrogen atoms within water molecules in our body. So you're using MRI when you have tissue with water in it. The hydrogen atom is the most abundant atom found in our body. 70% of our body is made up of water. So anything with water, which 70% of our body has hydrogen atoms. All protons in the atomic nucleus spin, which creates a small magnetic charge. So the proton is spinning. A magnetic field then aligns with the spin of hydrogen atoms, which you can see in the lower right, kind of like if you were spinning a soccer ball or a football or a basketball on your finger. Usually, they're all randomly oriented, but if you apply a radio frequency pulse that interrupts the protons to force them in a different alignment, it shifts their alignment by 90 degrees. So the hydrogen protons are spinning, and then they start to align in the magnetic field, and when you apply a radio frequency pulse. So for those of you who've had an MRI, that kunk, 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 kunk sound you hear in the scanner is when the radio frequency pulse is being applied, right? So what that's doing is shifting the spinning hydrogen molecules. Then you can turn that off, and it relaxes, and they all go back to their initial alignment, releasing their electromagnetic energy. Then Sometimes if you've had an MRI, if it's been a head, you have a little basket on your head, or if it's your knee, you might have something around your knee. That's a coil. What that does is it detects energy and can differentiate different tissue types based on the relaxation time. So tissue with the tumor in it has a different relaxation principle than tissue without a tumor in it. And as a result, you can start to see the contrast between the two, and you can start to pick up different densities of tissue using MRI. Okay, we're almost done with the physics. The decay rate of releasing the electromagnetic energy is different depending on the kind of tissue in the hydrogen atom, and this is how we build the MRI picture. So when we talk about white matter and gray matter, which we will do in just a minute, here's white matter and here's gray matter. It has different tissue properties, and the MRI allows us to distinguish those different tissue properties. There will be a test after this. I'll make it multiple choice, and I want you to write, or short answer, you can write a little discussion about how protons spin and they relax, and all right. So now that you know all about MRI and you're all an expert, how did we use MRI in the Bucharest Early Intervention Project? So we wanted to examine the anatomy of the brain in detail. We wanted to separate gray matter from white matter, which I'll define in just a minute. We wanted to examine the whole brain, as well as the gray and white matter volume. And we wanted to examine cortical thickness, because with development, the cortex should thin. So when I say cortex, when you look at a brain and you see those infoldings, the lumpy stuff that's the cortex. The cortex is just the outer part of the brain. If you strip that off, you have just the subcortical areas, all right? So the reason those infoldings occur, one reason is that the skull is fixed, and as the brain develops, it folds over onto itself. So if you look at the gyri, the bumps, and the soul side, the valleys, with the cellular layer there should thin with development based on experience. So cortical thinning. Remember we talked about how we overproduce synapses and we overproduce neurons, and then we prune them back? When they're pruned back, the cortex gets thinner. And differences in cortical thickness could be indicative of differences in how the brain absorbed experience, or in some cases, a lack of experience. So where I'm going with this is that you might anticipate that a child who's neglected and lacks experience would show less pruning and therefore maybe a thicker cortex. But we'll see. So we're going to start very simply and talk about gray matter and white matter. My fist is a part of a neuron, and this part is the cell body, and then I have little appendages that come off of it, all right, axons and dendrites. So anything in the brain that does not have myelin, I'll define myelin in a minute, turns up gray on an MRI. So things like dendrites don't have myelin, cell bodies don't have myelin, and some axons don't have myelin. But many axons do have myelin, and they show up as white on an MRI. And so what we can look at is the amount of white matter volume and gray matter volume. Think of gray matter as the central processor doing the computations. White matter is the information superhighway. Think of it as your phone line, if it's a landline phone, or if it's cell towers. It's what transmits information. So myelin makes a huge difference in how fast and how efficiently the brain works. But the gray matter is doing the math. It's the processor sitting in your computer. What we see is, on the left is when we scanned the kids when they were 8 to 10. On the right is when we scanned the kids when they were 16. You'll notice that both the children in the institutional group and foster care show a dramatic reduction in gray matter compared to the kids who've never been in an institution. And we see exactly the same finding when the kids are 16. So what we're seeing then is a reduction in gray matter. and no intervention effect. And this is very concerning because if gray matter is where the computations are being done, we've lost CPU power, which may well explain, as you heard before, why we see such deficits in IQ and executive functions. I have a brand new laptop, and it is so much faster than my last laptop. So think of, I have a much bigger CPU than I had before. Now, the next thing is, what about white matter? And now white matter is a little bit, I keep forgetting I have, is a little bit more encouraging. You'll see at both ages that although the never institutionalized children have much more white matter than the institutionalized, the foster care children are in between. So they have more white matter than they do, but less than they do. And then we see exactly the same thing at age 16. So we've replicated ourselves. So we see a small intervention effect for white matter, but not for gray matter. The other thing to note is we worried, well, what if we're not collecting our data correctly in Romania? So the typical pattern of development is as children move from pre-adolescent to into adolescence, there should be a reduction in gray matter and an increase in white matter. And if you look here, the thick black line is the never institutionalized children. And you can see that they go from a lot of gray matter to less gray matter. That's pruning. You'll see the other two groups do the same thing, but you'll notice that they have less gray matter than the never institutionalized group, which is as I just showed you. If you look at white matter, the expected pattern is you go from pre-adolescence to post-adolescence. There should be an increase in white matter, and we see the same thing. So what this is telling us is that our sample of never institutionalized children are showing exactly the expected pattern of development. A reduction in gray when they go from 8 years of age to 16, and an increase in white when they go from age 8 to age 16. All three groups show the same pattern, but again, we're seeing a difference by group. So in summary, at age 16, as at earlier years, we see a reduction in gray and white matter. At age 8 to 10, we see only a partial intervention effect in white matter, and we see the same expected rate of change from 8 to 16, with gray matter decreasing and white matter increasing, and the slopes differ by group. The next thing is I want to go to cortical thickness. So at age 16, these are preliminary data. We wanted to look at differences in cortical thickness in different brain regions for the reason I explained a little bit before. If the normal process is to thin the cortex, do we see differences in how thick the cortex is based on which of the three groups the children are in? So because it reflects pruning, a normal part of brain development is driven by experience, and with experience, the cortex should get thinner. So here's the way we can look at this. So this is showing us in one area, so you're looking at a view of the right hemisphere, looking this way, frontal lobe, and these red areas are highlighting the differences between the, in this case, the care as usual versus foster care. So this is, and what we're showing is that there are areas in the frontal lobe that are thicker in the care as usual than in the foster care kids. We can see that in this view as well. Again, everything that's in red is illustrating the difference in thickness, and it's favoring the care as usual or institutional group, meaning it's thicker for those kids than it is the foster care kids. This is a comparison of the never institutionalized kids and the care as usual kids. And again, we see it's thicker in the care as usual kids. Here's a comparison of foster care versus never institutionalized, and they have a slightly thicker cortex than the never institutionalized. So what that would tell us is that the cortex is thickest in certain areas in the institutionalized kids compared to the never institutionalized kids. And there are still a few areas in the foster care kids that are thicker than the never institutionalized. So the theme then would be incomplete recovery for the kids placed in foster care. And then the issue is that because of how old the children were when we placed them. Had we placed them at 6 months of age, maybe we'd see complete recovery. But since we didn't place them until around 20 or 22 months of age, maybe that's what's limiting their recovery. And you saw that also when Professor Fox talked in the IQ data, that we increased IQ, but we did not make the children in foster care group have a normal IQ. All right, so now let's turn to stress. We heard some of the very important findings Professor Fox talked about before, how children experiencing neglect have difficulties regulating their stress response. I'm going to highlight just one of these effects. So there's extensive evidence from both animals like rats and humans that early adversity, including neglect, can cause the body's stress response system to develop in an atypical fashion. Not having a normative healthy response to a stressor could, in theory, lead to the development of health problems. So we know people who are chronically stressed and show all the symptoms of chronic stress, physiological symptoms, are at a greater risk for developing health problems. So what we're arguing here is that exposure to stress early in life sensitizes you to respond in a bad way to stress later in life. So children who are experiencing chronic stress early may not be able to respond to a stressor later. But it may depend in part on also the severity of the stress and the number of stressors you're exposed to. So what we've done is we went back and we looked at the three groups, the institutional group, the foster care group, the never institutionalized group, and we kept track of the number of significant stressful life events each of the three groups experienced. And then what we wanted to know is, would we see an interaction between the number of stressful life events and your rearing conditions? So, for example, would the children in the institutional group who had a lot of stress have a different outcome than the children not in foster care or never institutionalized? And the other part would be, would this particularly be associated with what we call externalizing behavior problems like disruptive behavior or ADHD and things like that? And so what we're showing you here is the following. This is looking from the three different groups across time, and on the y-axis, the vertical axis, is the amount of externalizing behavior you're showing, like disruptive behavior, conduct disorder, things like that. And you'll notice that for the never institutionalized group and the foster care group, as the number of stressful life events increases, that's what the x-axis is, there's no difference. So these groups don't show any elevation in showing externalizing problems based on the number of stressful life events. A child with 7 stressful life events is really no different than a child with 1. But look at the care as usual group, the blue line. What that's telling us is that for the children in the institution, those who experience more stressful life events are much more likely to wind up with externalizing behavior problems. So that's the interaction. So it's saying there's a sensitization that occurs in particular to kids experiencing neglect. So children in the care as usual group are particularly sensitive to stress, that the more stressful events they report, the more likely they are to show externalizing behavior problems like acting out and disruptive behavior. And in contrast, the children placed in foster care and in the never institutionalized children do not show that sensitization to stress. So being in a family is protective against stress. And if you go back to Professor Fox's talk yesterday about the buffering effects of relationships, this supports that very thing. So lastly, now just a little biology and then I'll close. We wanted to know, over and above the effects on brain, how are these experiences of neglect biologically embedded? That is, embedded into your very biology, not just your behavior. So the way we wanted to look at this is how would we look at a biological effect? Well, we looked at something called telomere length, and I'll show this to you in just a second. So the length of the telomere region can serve as a readout of accelerated cellular decline. that's associated with the stress or adversity. So, if you look at the, I'm not very good with colors. Here's a chromosome. So all our DNA lives on the chromosomes. This is the telomere region. It's a cap. So every time your cell divides, the cap protects the cell. So if you cut yourself, or like in my case, I have a burn on my cheek, all right? You make new cells. You don't make new brain cells, but all the rest of your cells in your body are made anew. But they have to think of the fact that you go from being a baby to being a 90-year-old, and your cells over time start to not replicate as easily as they used to. And one reason for that is the telomere region gets smaller. And eventually, if the telomere region erodes enough, the cell jumps out of the cell cycle and doesn't replicate itself, which is what would lead to health issues. So we wanted to look at whether children exposed to profound neglect showed accelerated telomere erosion. So here's an example. Elizabeth Blackburn shared the Nobel Prize for co-discovering telomeres about 10 or 12 years ago. And she published a paper with her group at the University of California in San Francisco, looking at middle-aged women who were taking care of children with chronic disabilities. So imagine these are women taking care of kids with severe autism or severe other neurodevelopmental problems. And it turned out they had lost the equivalent of 10 years of telomere length because of the stress of taking care of children like that. So it's then led to the hypothesis that greater stress can accelerate telomere erosion, which then in turn could be associated with later developing health problems and things like that. So what we did is we looked over the first 16 years of life at the degree of telomere erosion. Now remember, everyone's telomeres get shorter over the lifespan. That's just the way development works. And actually the biggest decline is usually over the first 5 years of life. But everyone's telomeres decline over the life course. If you look at the blue line, those are the never institutionalized kids. And you see that they show a decline from infancy all the way through 16. But look at how much steeper the decline is in the institutionalized kids. They show greater telomere erosion. And then the next thing we wanted to look at is, so what? What does that mean? What are the implications? So the way we did that is since we can't get medical records in Romania, we asked parents or caregivers about the health of their kids. We basically, took a health record. I would say to you, is your child sick often? What kind of illnesses do they get? They get colds, things like that. And what we find is that on the far left are the telomere length among the children whose parents said they had excellent health. But notice on the far right, the telomere length of the children whose parents said their kids only had fair health is much shorter. So that's the health consequence. And then the last thing we wanted to look at was, is there an association with mental health? And what we see here on the x-axis is the degree of internalizing psychopathology like anxiety and depression. And on the y-axis, the vertical axis, is telomere length. And what you see on the far left is less, I know it's plotted funny, but you see less anxiety and depression the longer the telomeres. And as telomeres get shorter, you see more anxiety and depression. So previously we established health consequences. Now we have psychological consequences, that telomere erosion may mediate the response to increased anxiety and depression. So in summary, greater telomere erosion is we see among the institutionalized sample. At age 12, greater erosion is associated with more parent-reported health problems. Similarly, greater erosion is associated with more internalizing problems. So in conclusion, institutional rearing increases the risk for serious impairments and delays in multiple aspects of brain and biological development, that children removed from institutions in place on high-quality foster care show better brain and biological development, that earlier placement leads to more recovery, and that the quality of foster care matters, as does the stability of foster care. And on that note, I'll thank you for your attention.